Welcome! In this explainer, we're going to do a deep dive into Recirculating Aquaculture Systems, or RAS for short. We'll cover everything from the core principles and components to its real-world applications. This is designed to be a comprehensive study tool, so let's get right into it.
Okay, let's jump right into the single biggest reason RAS is such a game changer. Just look at these numbers. A traditional, old-school system might burn through 30,000 liters of water just to produce 1 kilogram of fish. A modern RAS, it can do the exact same thing with as little as 300 liters. That is a staggering difference in efficiency, and it's really the main driver behind this technology.
So, here's our game plan. First, we'll get a solid definition of what a RAS actually is. Then, we're going to trace the journey of water through a commercial system, piece by piece. We'll take a special look at the all-important nitrification process, recap the entire water cycle, and then finish up by exploring where and why RAS is used in the industry today.
All right, first things first. Before we can talk about the components, we need to answer the foundational question. What exactly is a recirculating aquaculture system?
You know, at its core, it's pretty straightforward. A RAS is a technology that lets you farm fish, or any aquatic organism, by taking the water they live in, cleaning it, and then sending it right back to them. The whole system is built around this continuous loop of treatment and reuse, which means you use way less water and have a much smaller environmental footprint.
Now for the fun part. Let's look under the hood at the physical components. And to really get a handle on how they all work together, we're going to follow a single drop of water on its complete journey through the system, from the moment it leaves the fish tank to the moment it returns, clean and fresh.
And that journey starts right here in the culture tank. Now, obviously, its main job is to hold the fish, but a well-designed tank does more than that. It's actually engineered to use water flow to concentrate all the waste, the feces, the uneaten food, and all the dissolved stuff like ammonia and CO2, and funnel it towards the exit pipe to begin the cleaning process.
So once that water, now loaded with waste, leaves the tank, its first stop is the mechanical filter. The industry standard is a rotating drum filter. Think of it like a big self-cleaning sieve. Water flows through a superfine screen that traps the solid particles. Then, as the drum rotates, powerful spray jets blast the collected gunk off the screen and into a waste chute. Its job is simple but crucial. Get the solids out and get them out fast.
Now here's a number you'll probably want to remember for an exam. That screen size is typically between 40 and 100 microns. And that's not just a random number. Being that fine is what allows the filter to grab even tiny particles before they have a chance to break down and pollute the water further. It's a critical detail.
Okay, with the big stuff out of the way, our water drop now moves to what I like to call the biological heart of the entire system, the biofilter. This thing is packed with special media, like little plastic bits called bioballs, that have an enormous amount of surface area. But this surface area isn't for physical filtering, it's prime real estate for billions and billions of beneficial bacteria to live on.
You know, this bacterial process is so unbelievably important, it's the engine that makes the whole system run. So we need to pause our water's journey for a second and zoom in on it. It's called nitrification, and without it, reusing water on this scale would be impossible. So here it is. This is the magic formula, really. Fish don't just produce solid waste. They constantly excrete ammonia, which is highly toxic, straight from their gills. Inside the biofilter, one group of bacteria takes that toxic ammonia and converts it into something called nitrite. The problem is, nitrite is also very toxic. So then a second group of bacteria comes in and converts that toxic nitrite into nitrate, which thankfully is much less harmful to the fish. And this isn't just one step, it's a two-part process, and you've got two different teams of bacteria doing the work. In step one, you have ammonia oxidizing bacteria, or AOB. A good example is Nitrosomonas, and they handle the ammonia to nitrite conversion. Then, in step two, the nitrite oxidizing bacteria, or NOB, like Nitrobacter, take over and finish the job, turning nitrite into nitrate. For the system to be stable, you absolutely need both of these colonies to be thriving.
But here's the catch. A brand new biofilter is basically a blank slate. It's sterile. It can't do this job from day one. It actually takes anywhere from 1 to 3 months for these bacterial colonies to grow large and strong enough to handle the waste load from the fish. You can sometimes speed this up by seeding the filter with bacteria from a mature, established system, but it's a process that takes time and patience.
All right, let's get back to our drop of water. It's just left the biofilter, where not only the fish, but also those billions of bacteria have been breathing, producing a ton of carbon dioxide. So the next stop is a degassing tower.
Here, the water trickles down over media, which creates a lot of turbulence. This action effectively strips out those unwanted dissolved gases, mostly CO2 but also others like nitrogen or even toxic hydrogen sulfide, and releases them into the air.
So we've taken the bad gases out, and now it's time to put the most important gas back in, oxygen. This is often done with an oxygen cone, which is a super efficient device that injects pure oxygen into the water under pressure. The whole point is to bring the oxygen saturation level all the way back up to 90 or even 100%. This is absolutely critical for keeping fish healthy and growing fast, especially at high densities.
And just like with the drum filter, here's another key technical number to file away. These oxygen cones typically operate at a pressure between 1.5 and 3 bar. It's that pressure that forces the pure oxygen to dissolve so efficiently into the water, making it far more effective than just bubbling air through an air stone.
We're almost at the end of the treatment loop. This is the final biosecurity checkpoint, disinfection. There are two main ways this is done. One is with ultraviolet, or UV light. The water flows past a powerful UV lamp that zaps any pathogens and damages their DNA so they can't reproduce. The other method is using ozone gas, which is a powerful oxidizer that basically obliterates pathogens and other fine organic particles on contact.
And at last, our fully cleaned, oxygenated, and disinfected water arrives at the sump. The sump is basically a collection tank. It acts as a buffer in the system and gives any last tiny particles a chance to settle out. From here, a powerful pump gives the water the push it needs to travel all the way back to the fish tanks, completing the cycle.
Okay, we've walked through each piece of the puzzle. Now let's take a step back and look at the entire flow from start to finish. This is a great way to review the whole process.
So here it is, the complete cycle, laid out step by step. It always starts in the fish tank, then goes to the drum filter for solids, the biofilter for dissolved waste, the degassing tower to remove CO2, the oxygen cone to add O2, and then through disinfection. Finally, it all collects in the sump before the pump sends it back to the beginning. And just a keynote, if you're working with a saltwater system, you'll almost always see a protein skimmer in the loop, usually right before the sump.
So we've spent a lot of time on how a RAS works. Now let's talk about why it's so important. Where do we actually see this technology making a difference out in the real world?
And the answer is, well, in a lot of places. The applications are incredibly broad. You see it in everything from large-scale food fish production and integrated aquaponics farms to highly sensitive hatchery operations, the farming of specialty high-value species, and of course, in research and education.
But let's make this more concrete. For food fish, RAS makes it possible to grow species like tilapia at high densities in places where land or water is scarce, like right in the middle of a city. In aquaponics, that fish waste becomes a valuable resource, fertilizing crops like lettuce or herbs in a single, dual-income system. For hatcheries, the biosecurity is a massive advantage. You can protect fragile baby fish from disease, ensuring a steady, year-round supply of fingerlings and cutting down on the need for imports. And finally, for science, the total control you have in a RAS makes it the perfect laboratory for precise research on nutrition, genetics, or for training the next generation of technicians.
So I want to leave you with a thought to chew on. Given that a RAS gives you the power to create a perfect, controlled environment with incredible biosecurity, what does that mean for the future? What new, high-value fish or other species, things that maybe aren't common in your local area right now, could this technology suddenly make possible to farm? It really opens up a world of possibilities.